All right, this is chapter 5 of your textbook, large biological molecules or macromolecules. This is part 2 where we talk about proteins and nucleic acids. And the last one, we kind of did a general overview and we talked about carbohydrates and lipids. All right, here we go. Proteins account for more than 50% of the dry mass of most cells. Proteins make us who we are, which is why we spend a whole unit talking about protein synthesis. Enzymes are proteins, and they speed up chemical reactions in cells. And remember, still in this unit, we will be talking more specifically about enzymes. Proteins are constructed from monomers called amino acids. Remember that all macromolecules are made up of simple monomers. And in this case, the amino acids. And remember, we also studied a dehydration reaction as well as a hydrolysis. Really quickly, see if you can remember what happens in a dehydration reaction and what happens in a hydrolysis reaction. Okay, you need to pause the video. Go ahead and pause the video while you finish that up. But I'm going to tell you, dehydration reaction actually builds things. It synthesizes. So when you're taking the monomer amino acids and building a protein, that's a dehydration reaction. And we actually lose a water in that process or form a water. Hydrolysis reactions are going to break down polymers into monomers. So this is where we actually are taking proteins and breaking it down into the simple amino acids. So, for example, maybe if we ate a steak, our bodies would digest those proteins and break them down into amino acids. The hydrolysis reaction actually requires a water to be part of that. So amino acids are structurally all the same except for the R group, which is an area that is different in all the 20 amino acids. So they all have the basic same structure except that R group. That's where they all differ. Page 79, figure 5.6 shows all the amino acid structures. You don't have to memorize all the amino acids, by the way. Page 78 outlines some of the jobs that those proteins have. So here are some of the categories of jobs. Enzymes, those help with chemical reactions. Antibodies are examples of defense proteins. They help defend the body with like the immune system. There are proteins that are for storage, proteins for transport, proteins that are receptors on cells, humoral proteins, contractile motor, and structural proteins. So there's lots of jobs that proteins do. Because remember, more than 50% of cells, of the dry matter cells, which doesn't include the water, obviously, is proteins. If that's what makes us who we are. So here are some of those functions. And again, this is in your textbook. So enzymes, remember, speed up chemical reactions. And we'll do a whole section in this unit on enzymes. Defense proteins, think about antibodies. They're actually going to help protect your body. Storage proteins, casein is a protein in milk. It's a major source of amino acids for baby mammals. Plants have storage proteins in their seeds. Oval human is the protein in the egg white, and that's used as an amino acid source for the developing embryo. Transport proteins, hemoglobin transports oxygen in the blood. Let's see. Transport proteins can also be embedded in the cell membrane, and those help move molecules across. Humoral proteins, coordination of an organism's activities. So insulin is an example of, I said humoral, I meant hormonal. Sorry, I can't read. Insulin is a hormone that is secreted by the pancreas, which allows the cells to take up glucose. Remember, people that have diabetes don't have insulin, and so therefore they can't take that glucose up in their bodies, and so their blood remains really high with that sugar, and then that's bad if you have high blood sugar. Receptor proteins, these are proteins, so you can find them on the cell, and they actually accept things like neurotransmitters or other signaling molecules that are released by other cells, and they actually recognize that and take it up into the next cell. Contractile motor proteins, a good example, this would be actin and myosin. These two are important in movement of your muscle tissues, your skeletal muscle tissues. So here's the myosin fibers and here's the actin. And the myosin actually links up with the actin and it actually causes it to contract, to pull together. It'll pull these two parts inward together, and then as it releases, it will release them back out. Structural proteins. Keratin is in your hair. It's in horns. Obviously, you don't have those. Feathers or other skin appendages. Insects and spiders use silk fibers to make their cocoons and webs. Collagen and elastin also provide framework for the connective tissues in our bodies. When two amino acids join, they combine with a peptide bond, okay, by a dehydration reaction. Remember, dehydration reaction removes a water when it happens. Who remembers what a dehydration reaction is? I hope at this point you're saying, me. Proteins have primary, secondary, tertiary, and sometimes quaternary structures. The final 3D shape is what determines its function. Remember, back in Unit 2, I said shape equals function. And even a small change in the shape can distort the function of the protein. We'll talk about an example of that here in a little bit. Page 82 and 83, figure 520, shows the 4 levels of structure. The function of the protein depends on its ability to recognize and bind to some other molecule. So again, remember, the shape matters because if it's not shaped correctly, it's not going to bind to another molecule. Many diseases are associated with misfolded proteins. So let's show how important that structure is. Sickle cell disease is due to one amino acid change in the primary structure, which affects the secondary and tertiary structures and causes red blood cells to form a sickle shape, like this, instead of like that. And that actually clogs the blood vessels. So these are the amino acids, and there's lots of them. You do not have to really know them. This is that dehydration reaction. Peptide bond forms between them. Notice this is the R group here, okay, of an amino acid, and then the rest of the amino acid is basically all the same. Okay, it's just the R group here that differs in any amino acid. So here's a ribbon model of a protein, and this is a space build, where you don't see all that space. This shows how that primary structure is then woven into a secondary and tertiary structure. So primary structure is right here. So this is your primary structure. It's the actual sequence of amino acids. Okay, that's what your primary structure is. Then your secondary structure is how that sequence is actually folded. You've got beta pleated sheets or an alpha helix. It doesn't matter that you know those. Understanding that it's taking this primary structure here and it's folding it into something more complex. Then the tertiary structure is when you take those folds, then how do you 3D it? How do you turn it into something 3D? Quaternary structure, not all proteins have this, but that is basically taking one protein here and another protein here and putting them together and actually making one large protein. So it actually is taking polymers and combining them. So here's that sickle cell anemia we were talking about. So notice that the 6th position of the amino acid, in a normal person, it's glucine. In a sickle cell person, it is valine. Well, what that does is that actually distorts the structure. Notice this is a normal and this is not normal. And because of that, when it combines with other proteins, so here's multiple proteins here, the 2 of these are distorted. And what that does is that causes the sickling shape in the red blood cells. This is a normal red blood cell. And that affects the capacity to carry oxygen. And you can think about the importance of carrying oxygen to all of your body and all of your muscle cells. Well, if you're not carrying oxygen to them, well, that's a problem because you're not going to be able to do very much. Your cells need oxygen. Cellular respiration requires glucose and oxygen. Your cells aren't going to do efficient cellular respiration if you don't have oxygen, which means you're not going to produce a lot of ATP. Oops, ran out of writing space there. And if you're not producing a lot of ATP, you're not going to be able to do much. You're going to be low on energy. Denaturing a protein can happen when the protein becomes biologically inactive. Interactions within the protein structure are destroyed and the shape is not functional. I should say no longer functional. It can result from excessive heat, chemicals, pH levels, being out of range, or many other things. DNA is another thing that is a chemical compound that is actually a nucleic acid, but it can also be denatured. You can denature any kind of protein or nucleic acid, and that means like if you heat it excessively, or you add chemicals to it, or you whack its pH level, it can actually break down that protein. Where is it breaking it down? It can break it down at the primary level, where these amino acids are no longer linked, or it can break it down here. And all of a sudden, this pleated structure, or this helix, these pleats, those don't form, and then it can't link up with other ones, and so it can really throw that entire protein out of whack. And when it does that, it's what we call denaturing. It breaks down that protein structure. Body temperature is a great example of this. Excessively high fever will actually cause a multitude of enzymes within the cells of your body to denature, and death can occur. Please turn it down. Sorry, that was not directed to you, that was directed to my kids. So here's our normal protein. When we denature it, it actually loses the structure. Right? Can it be renatured? Yeah, possibly. Not often in nature. Once you destroy it in your body, sometimes they're just destroyed. Determining the 3D structure of proteins is not easy. We have X-ray crystallography. That has helped us a little bit in figuring out the protein structure. Or magnetic resonance spectroscopy, NMRs. We've developed some of those, but there's still a lot of unknowns. Okay, this is really where it's weird. There's just a lot of structures of proteins that we don't know. There's a program called Foldit. Feel free to Google it. But it's actually developed by scientists, and it's like a game. And you sit there and you try to figure out the 3D structure of proteins, because scientists can't do it all alone. And so they created this protein out there. We were actually playing a game to figure out if the structure fits, and if it could actually do anything with that structure. And we've actually found a lot of protein structures that might actually fit. That all this data is sent to scientists, and then they can look more closely at, well, would that structure actually fit? So just keep in mind, we don't know all the structures. The future of determining these structures may help us cure diseases like Alzheimer's and Parkinson's. Remember, shape gives rise to function. So if we know that there's a protein in Alzheimer's that is no longer operating properly, we can actually manufacture a chemical compound or something that mimics the protein that is no longer functioning. We've already done that with diabetes and insulin. We have manufactured insulin so that the body, you get that insulin, and the body actually recognizes it as insulin, even though it's not your body-made insulin, it's man-made insulin. And we now have a way to control diabetes, whereas people died of diabetes 100 years or so ago because we didn't have that. So we're looking at it. It's really interesting. So nucleic acids are things like DNA and RNA. They're made up of nucleotides. Those are their monomers. DNA and RNA allow organisms to pass information from one generation to the next. This is what their job is passing information. The flow of genetic information, which we have a whole unit on this, is DNA, RNA, and protein. So we first have DNA, which is transcribed into something called RNA, which then builds the protein. And actually what it builds is it builds amino acids, but amino acids build a protein, because remember those are monomers for protein. Nucleotides consist of three parts. The nitrogen-containing base, a 5-carbon sugar, and a 5-carbon sugar is drawn like this because it has 1, 2, 3, 4, and 5 carbons. And one or more phosphate groups. DNA is double-stranded, RNA is single-stranded. They run anti-parallel. So one end is 5' and one end is 3', and the other is just the opposite. We're going to learn more about that when we get to DNA. I think I have a picture here, and I can show it to you here in just a minute. DNA carries your inherited information in the form of something called genes, which is a sequence of your bases like A, T, G, and C. And that determines the overall amino acid sequence and thus the protein structure. So that's all protein synthesis. But the big idea here is that the order of these bases creates a sequence of amino acids. And that sequence of amino acids determines what the protein is. For example, if you are light-skinned, you have less protein called melanin. Well, how does your body determine how much melanin to make? It's based on the sequence of your genes. And the sequence of your genes will determine what amino acids are present, which will then determine whether you have more melanin or less melanin. So this is what we mean by 5' and 3'. So this is your sugar. And then what they do is they count these as carbons. So this is 1, 2, 3, 4, and 5 carbons. And so this is the 5' and here's the 5th carbon. The other end is just the opposite. This is the 1, 2, 3, 4, and 5 carbons. And so, oh wait, that's not a carbon, sorry. 1, 2, 3 carbons, 4 carbons, and this is a 5th carbon. I was counting on 1, 2, 3, 4, 5 up there. So this is the 3' end because it has that 3rd carbon attached to it. And then if you built another DNA molecule on the other side, so where it's double-stranded, it would be a 5' end here and a 3' end here. That's what we mean by anti-parallel. And that means that this side is right side up and this side upside down. And this is a nucleotide, so it's a phosphate, a sugar, carbons 1, 2, 3, 4, and 5 carbons, and then a nitrogen base. And the nitrogen base can be cytosine, thymine, adenine, or guanine. Now, when it's RNA, there's no thymine, it's uracil, and we have a chapter on that later in the year. So this is a picture of DNA here. So the bases are joined by hydrogen bonding. Remember, hydrogen bonding is kind of a weak bond. RNA and DNA are joined by that. Let's see, 5' and the 5' and notice the other side is just opposite of it. This is just showing the thread here. So this is actually the sugar and the phosphate. Sorry, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. See that background. Okay, and that's it. That was proteins and nucleic acids. And we'll be working with all of those proteins and nucleic acids more so when we get to Unit 4.